All right, this video is going to cover Chapter 8, Part 2. We're going to talk about glucose metabolism, how bacteria make ATP. So again, there's two components of metabolism. We mentioned enzymes were involved in that. There's catabolism or catabolic, which is a breakdown of the complex organic molecules into simpler compounds. And here we're going to get a release of energy, meaning that we're actually going to make energy. So just because you're breaking something down doesn't mean you actually use energy, but you should have a net overall gain of ATP. Anabolism or anabolic, we're going to build complex organic molecules from simple ones and require energy to do so. So energy is basically the capacity to do work. There's two forms. There's potential energy, which is stored energy, and kinetic, which is energy in motion. So example, rock on top of a hill is potential energy, and when that rock is tumbling down the hill, that is kinetic energy. So ATP is the energy currency of the cell. So it's a donor of free energy. There's 3 phosphates associated with ATP, and energy is stored in the phosphate bonds. So I like to think of ATP as kind of like a charged battery. Everybody's familiar with a battery. You have them in your cell phones. And when your battery in your cell phone is fully charged, it's full of ATP, so to speak. Now there's a cyclical relationship with ATP and ADP. So there's only 2 phosphates with ADP, and I like to say this is the dead battery. So when the battery on your cell phone runs out, it's got nothing left but ADP. It needs to be charged, make more ATP. So here's a little schematic here showing that cyclical relationship with ATP and ADP. Catabolism, here we're going to store energy. Anabolism, we're going to make larger macromolecules, and that ATP is used for other cell processes like cell division and things like that. So cells require a constant, they constantly generate and use ATP to power biosynthetic reactions. The 2 processes used by bacteria to form ATP are substrate level phosphorylation and oxidative phosphorylation. So substrate level phosphorylation is a release of chemical energy used to add a phosphate to ADP. So our example here is going to be glycolysis and the Krebs cycle. And the thing with substrate level, so substrate, think enzyme, food, chemical energy, only a small amount of ATP is actually made. We're going to get most of the ATP produced by what's referred to as oxidative phosphorylation. And this is harvesting energy from what's called the proton motive force. The proton motive force is produced by the Electron Transport System, and we will get to this later. So we need to have a chemical energy source, so an energy source or electron donor, because what's going to happen is glucose is oxidized when it's broken down. So it's going to lose electrons. Oxidation is a loss of electrons. So that's what's happening to glucose. When you have oxidation, you have to have reduction at the same time. So these reactions go hand in hand. So reduction is the gain of electrons. So I like to think oil rig, oxidation is loss, reduces gain. So what's going to happen is glucose is oxidized, and we're going to have an electron acceptor pick up the electrons, and those electron acceptors are going to be reduced. So like I said, these oxidation reduction reactions have to occur at the same time. We have to have an electron transfer from a donor to an acceptor. So our donor in our example is going to be glucose. Our acceptor is going to be NAD+, which is an electron carrier. These reactions always occur simultaneously. Cells use these electron carriers to carry electrons and often hydrogen atoms. And the electron carriers I will refer to as reducing power because these electrons that they pick up actually have energy, and we can harvest that energy from those electrons later. So we are concerned with 2 types of electron carriers. There's NAD+ and FAD. They have long chemical names. It's not important. But when they become reduced, they pick up electrons from glucose. Our reduced form is NADH and FADH2. Okay, so when glucose is oxidized, glucose loses electrons. These are the form in which they pick them up, and this is what they become when they do pick up those electrons, and as you see, hydrogen as well. These electron carriers, like I said, I call these reducing power because they power the electron transport system, which will create what we call the proton motive force. Ultimately, these electrons need to go to a terminal electron acceptor. So it's a molecule such as oxygen that these electrons can attach to. So in aerobic respiration, oxygen is the terminal electron acceptor. There's other types of respirations like anaerobic respiration, where something that's inorganic other than oxygen is going to pick up the electrons. So nitrate or sulfate, for example. So looking at glucose metabolism, microorganisms will oxidize carbohydrates as a primary source of energy for their anabolic reactions if they can use the actual carbohydrate. So in the lab, we do tests for glucose, lactose, mannitol, fermentation, just for a couple examples. Some organisms can use those sugars, others cannot. If they can use those sugars, they will use that as a primary source of energy as long as it's available. Meaning, just like your cells will use carbohydrates as a primary source. They're going to hold off on breaking down proteins and fat until that sugar, glucose, is used up. Like I said, there's different types of respiration. There's aerobic and anaerobic. And then there's a non-respiratory metabolism, which is called fermentation. And we'll briefly cover that later as well. So looking at the key metabolic pathways that we'll look at here, we're going to gradually oxidize glucose to basically break it down completely to carbon dioxide. So glucose is a 6-carbon sugar, and we're going to break it down into basically carbon dioxide and water. So we're going to look at glycolysis, the transition reaction, also referred to as the synthesis of acetyl-CoA, Krebs cycle, and the electron transport system. We're going to follow this general chemical equation, C6H12O6 plus 6 oxygen. Oxygen is the terminal electron acceptor. We're going to yield carbon dioxide and 6 waters. In this process, this is bacteria again, we're going to get 38 ADP plus 38 phosphates, making 38 ATP. So glycolysis, if you were to describe glycolysis to somebody, basically what you could tell them is it's a sugar splitting reaction. It's a 10-step process. You just need to know the basics here, what goes in and what comes out. So the input in glycolysis is glucose, a 6-carbon sugar. And we're going to split it into 2 molecules of pyruvate, which are 3-carbon. Okay, so if we count the carbons, 3 times 2 equals 6. All right, so we have 2 3-carbon molecules. In order to split glucose, we have to use some energy. 2 ATP are invested. However, we are going to have a payoff phase. We're going to generate more ATP than what we actually use. So we're going to generate 4, but since we used 2, we're going to have a net gain of 2. Okay. In order for metabolism to work, you have to have a net positive gain of ATP. Otherwise, it's you're not going to last that long. The end products of glycolysis are 2-pyruvate or pyruvic acid, which are the 3-carbon molecules. You're going to get 2 NADH and 2 ATP. Okay, so that's the net gain. The NADH is the reducing power that's created when glucose is oxidized. So glucose is going to be broken down, it's oxidized, and those electron carriers, the NAD+, are going to be reduced and make NADH. This is a schematic of glycolysis. It's a 10-step process, like I said. You luckily do not have to memorize this 10-step process, but if you look at the blue circles here, there are 6 of them. This is glucose. You can see where ATP is used, and then you're going to have ATP gained, and eventually have pyruvate, which are the 3-carbon molecules. Okay, so since it's split, you get 2 ATP for for the formation of one pyruvate, and 2 ATP from the formation of the other pyruvate. Now, after glycolysis, what happens is if the cells can respire, there will be 3 other reactions that take place. So this includes the synthesis of acetyl-CoA, or the transition reaction, like I mentioned before, Krebs cycle, and the electron transport system. So the electron transport system can be either aerobic or anaerobic without oxygen. So looking at the transition reaction, this connects glycolysis to the Krebs cycle. Okay, we're going to have an input from glycolysis. So these are connected pathways here. We're going to have an input of 2 pyruvate, and the end products are going to be 2 acetyl-CoA enzymes, 2 CO2, and 2 NADH. So here's pyruvate feeding into this system. We're going to have NADH formed. We're going to have CO2 given off. We're going to have acetyl-CoA created. Acetyl-CoA is a 2-carbon molecule. We lose that carbon from one pyruvate as one of these 2 CO2s. So since we have 2 pyruvate going in, we're going to have 2 carbons lost, 2 carbon dioxides produced. So this is a little schematic showing you just for one pyruvate. All right, so if this is the 1 carbon loss, that's 1 CO2 and 1 NADH. So you multiply that by 2 since there's 2 pyruvate. Now what's going to feed into the Krebs cycle is acetyl-CoA. And this is a cyclical process. Again, since we're dealing with 2 molecules of acetyl-CoA, it has to quote-unquote turn twice for 1 molecule of each of those acetyl-CoA. So input is acetyl-CoA, and the overall products we're going to get 2 ATP, 6 NADH, 2 FADH2, and 4 CO2. Okay, so we lost 2 CO2 in the transition reaction. Here are the other 4 CO2s created, thinking back to that equation where we had the 6 carbon dioxide created. So at the end of the Krebs cycle, substrate level phosphorylation, remember the chemical energy, this process is now over. And if you look here, we had 2 ATP in the Krebs cycle. Remembering back to glycolysis, we had a net gain of 2 ATP. So overall, substrate level phosphorylation, you create 4 ATP. Krebs cycle schematic, again, this isn't not important for you to remember, but it does show you where NADH is being created. NADH, so for 1 acetyl-CoA, you get 3 NADH, and you get 1 FADH2. You get 1 ATP. The second acetyl-CoA feeds in, again, you get the 3 NADH, 1 FADH2, and the 1 ATP. Now, probably the more complex portion of this chapter would be understanding the electron transport system. It's not everybody's favorite topic, but it really isn't that too bad if you put a little bit of time in. So our input into the electron transport system is the reducing power, so the NADH from the glycolysis and transition reaction, and the NADH and FADH2 from the Krebs cycle. Sorry, this should be a typo. This is the Krebs cycle. It's also referred to as the TCA cycle, but I refer to it as the Krebs. So anyway, the reducing power feeds into this system. Now the electron transport system in bacteria occurs across the cell membrane. So the electron carriers, or the reducing power, carry electrons to the cell membrane. So I'm going to pause here, go to my writing board, do a little demonstration again. Now, here I'm back on the writing board. I have 2 lines here representing my cell membrane. And the electron transport system, basically it's a series of proteins referred to as cytochromes in the cell membrane. And one special one, I'll get to it eventually. So here's my cell membrane. These are my cytochrome proteins. And what's going to happen is the NADH is going to give electrons give electrons and lose hydrogen to these cytochromes. Okay, so NADH becomes oxidized, loses electrons, loses the hydrogen, and eventually will follow these electrons through the system, but the reducing power donates the electrons to the cytochromes. This electron energy is shuttled from a cytochrome to cytochrome, and it's used to eject protons or hydrogen ions from the cell membrane. And this is what's going to actually form the proton motive force. So as these electrons are shuttled from electron carrier to electron carrier, they'll eventually come back into the cell. We'll get back to that later. But as a result, the energy of the electrons pump out the protons. And what happens is, since we have so much of the reducing power, we have a lot of protons that are pumped out of the cell. So my protons here, H plus, pumped out. And what happens is we get a net charge outside of the cell membrane as a positive charge. Inside the cell, we have a net charge of a negative. So think of something that has a positive charge and a negative charge, something you might use daily. If you thought a battery, you would be correct. A battery is potential energy, and that's what we've created here with this proton gradient. Okay, we have this electrochemical gradient or concentration gradient. Okay, outside of the cell, and you should mostly all be familiar with concentration gradients. Concentration gradients like to go from a high concentration to a low concentration. So there's a natural attraction because of the electrochemical charge here and the concentration gradient for these protons to go back into the cell. However, they cannot do that because they are not permeable to the cell membrane. So they actually have to be let back into the cell through this last enzyme here. It's called ATP synthase. This is a spinning turbine enzyme. This thing actually moves and it is powered by those protons flowing through ATP synthase. So those protons, they come back in. They will form with combined with the electrons and the terminal electron acceptor oxygen, and they will make that water that we talked about in that equation. Okay, this is not a balanced equation by any means, but this is in a chemistry class. But eventually we will get water that form. But ATP synthase, it will phosphorylate those 38 ADP and make the 38 ATP. It's the spinning action that powers this reaction. Okay. So, when those electron carriers transfer electrons, the electron carriers are recycled. So NADH becomes oxidized to NAD+. It goes back to glycolysis, Krebs cycle, picks up more electrons, repeats this process all over. Aerobic respiration is going to ultimately pass the electrons to oxygen, as I demonstrated. If it's anaerobic respiration, it's going to go to a terminal electron acceptor different from oxygen. Like I mentioned, the electron transport system creates the proton motive force. So what is the proton motive force? Since that lipid bilayer is not permeable to those protons, those positively charged protons build up outside of the cell, and they're concentrated immediately outside of that cell membrane. And the separation charges that it creates, like I mentioned, the electrochemical gradient, is potential energy. Okay, so since those protons have that natural attraction back into the cell, It's potential energy. Just like a battery. Once you put that battery in a flashlight, you connect the circuit with the button, those electrons flow. Okay? Here, our potential energy is harvested when the protons flow through ATP synthase. So what is the proton motive force? The proton motive force is potential energy created by this separation of charges. It's created by that concentration gradient. It's created by the electrochemical gradient. Okay, so if you're familiar with concentration gradients, you can understand what the proton motive force is. It's just the potential energy. And that potential energy is harvested when those protons flow through those ATP synthase enzymes. And that's used to phosphorylate ADP. There's an image in your textbook. It's much simpler than this, but it gives you a more accurate representation of what's happening. Here you see the NADH being oxidized, following the electrons, coming back in, combining with the oxygen, and those protons, the hydrogen, making the water. But you see here, the protons are pumped out, and they flow back through ATP synthase, and here we make our ATP. This process is done in your cells inside the mitochondria. Again, bacteria cells, it's going to be in that cytoplasmic membrane, the cell membrane, across that membrane. Now, how do we get 34 ATP from the electron transport system? So we get 3 ATP for every NADH and 2 ATP for every FADH2. So you get more ATP from NADH than FADH2, and I can kind of explain that here. NADH gives electrons right away to this initial cytochrome, which results in hydrogen being pumped out right away. FADH2 doesn't donate electrons until this second cytochrome, and eventually will pump out hydrogen. So NADH, you're going to get protons pumped out right away, and FADH2 is going to be delayed a little bit. So you get more ATP for the NADH. So going back to how much ATP we create, the NADH we get 2 ATP or NADH from the glycolysis, 2 from the transition reaction, 6 from Krebs cycle, for a total of 10. So 3 ATP per NADH, 3 times 10 is 30. For FADH2, we didn't get any until Krebs cycle. We get 2 ATP for every FADH2. 2 times 2 is 4. So 30 plus 4, 34 ATP. Now remember, we got 4 ATP from glycolysis and the Krebs cycle. So 34 plus 4, 38 ATP theoretical yield for 1 molecule of glucose. This is aerobic respiration. Aerobic respiration. Aerobic respiration, again, oxygen is used as a terminal electron acceptor. We're going to get the most amount of ATP yield when we have oxygen as the electron acceptor. Anaerobic respiration, inorganic ions, molecules other than oxygen are used. It's less efficient, less ATP is generated. So anaerobic is always less ATP. I'm not going to go through an example of anaerobic respiration. It's not important for us, but you should know that it's always less ATP. Now, switching topics to fermentation. Fermentation is done by organisms that cannot respire. So they don't have an electron transport system. So these organisms, again, they're indifferent to oxygen, meaning they can survive with or without oxygen, but it does not inhibit the growth. So meaning oxygen is not going to be toxic to them. It just does not use oxygen as a terminal electron acceptor because it doesn't have an electron transport system. But during fermentative metabolism, organic molecules are going to act as electron acceptors, but it's an electron acceptor that is going to be involved in recycling NADH without the use of an electron transport system. So thinking back to facultative anaerobes can switch their metabolism. So in general, in our lab, we say that we use these facultative anaerobes, but in fact, they could actually be fermenting. We just wouldn't have enough information just using one or two tests. So what happens during fermentation is that cells, basically, they can't completely oxidize glucose. Okay, so the complete oxidation of glucose occurred using the transition reaction and Krebs cycle. Glucose was lost as carbon dioxide. Okay, fermenters do go through glycolysis, and they form the 2-pyruvate or the pyruvic acid, which are the 3 carbon molecules. So you get 2 of them, you get 2 ATP, 2 NADH, and the next steps vary based on the organism. So depends on the organism and the electron acceptor. It's all used to recycle NADH. CO2 is usually always a byproduct. Okay, but I'll give you an example here with alcohol fermentation. We have glucose going in to the system. It goes into glycolysis. We get the pyruvate, and the next step here is basically the recycling action. We're going to get CO2. We're going to get recycling of NADH. Okay. This isn't important to you guys at all, but it does give you the schematic of what is happening. But when you lose the 2 CO2 from the pyruvate, you were left with a 2 carbon molecule of ethanol in the alcohol fermentation process. Now, again, depending on the organism and the electron acceptor during that recycling step here at the bottom, we get different products. So here's some example: Propionibacterium. These are involved in making Swiss cheese. So we get the propionic acid and CO2, and those pockets of holes that you see in the cheese are from the CO2 that's produced during the fermentation process. Lactic acid from your lactobacillus and streptococcus, used to make cheddar cheese, yogurt, soy sauce. Saccharomyces cerevisiae, which is a yeast, going to produce CO2 and ethanol. Obviously, our wine and beer, liquors, all from a fermentation process. Clostridium species are used to make acetone and isopropanol, so some organic solvents. All right, so this is going to conclude the second chapter 8 video. Make sure you go through the study guide, and if you have questions, make sure you let me know before you take the online e-learning quiz.